Yes?
Yeah.
Very good. Okay, so welcome. And it's a particular pleasure for me to have Leonardo Cerliani today talking at our clinical neuroanatomy seminars. Cerliani is not only a friend, is someone who got a very interesting career. He started studying philosophy.
Yeah.
Yes? I always wanted to do philosophy, but I didn't manage to. But then he thought perhaps philosophy was too vague, and he went into neuroscience.
Actually, I already went into neuroscience when I was doing philosophy.
So the two things were linked in his mind. He did his PhD in Turin, where the great Luigi Rolando was working before him 150 years ago. And Moss also, so great Italian anatomists came from that school. Then he moved to Amsterdam and Groningen as a postdoc, and now he's continuing his postdoc in Paris with Michel Thibault de Schotten. Now, Leonardo didn't want to reveal that much what is behind that title, structure or brain connectivity. It could be, everything about the brain, but I guess he will limit himself to an overview of the methods that can be used, have been used, or will be used to look at connectivity of the brain.
Yeah.
Thank you, Leonardo.
Thanks to you. Well, you can imagine that I'm a little bit emotional because the perspective of giving a talk at the IOP is quite emotional for everybody, even though I already gave a talk 2 years ago, but it's still emotional. I want to start with a question to you, because it's about clarifying potential linguistic ambiguity. In England, you had Wittgenstein for a long time, and made clear that many problems in language actually come from the fact that people use the same word but they intend different things. So my question is whether the word structures related to the brain makes you think about something which is opposite with respect to function. I would like to, it works in Italian, but maybe in English is different. I want to make sure that we are using the same word with the same meaning. So for instance, there is a journal, a very beautiful one, called Brain Structure and Function. Why do you think that they specify structure and function in the same title? What do you mean in English when you talk about structure? What do you think when you think about structure of connectivity in the brain? It's a real question, I wait for an answer.
Something is more static, at least have a
okay.
static temporality.
Yeah, the physical appearance of something.
Okay. Okay, when you think about structure, you think about the functional connection.
Architecture of the brain.
Okay, can you define that better?
Yeah, like the composition of it, like how everything is connected, not necessarily functions.
Okay.
I kind of think they're two separate. They are together intrinsically.
Fantastic. Other ideas? We can stay with this. Okay, now I see that the same ambiguity is there in Italian and in English. In the sense that indeed you can think about structure as something like the hardware, something that stays underneath, but actually it doesn't do that much if not just allowing something to happen at the functional level. Or you can think about structure in terms of internal organization, like the architectonic organization of a building. That is something which is in a certain way static, but it represents the organization of the building itself. I will use, every time that I will use the word structure, I will intend this second meaning. So I do not intend to oppose structure to function. I will use the term structure to describe what we can do when we consider the connectivity of many different voxels, many different parts of the brain, and try to look at patterns in that large range of connectivity descriptions. And I will start with this slide that most likely you already know because it's from Michel Thibault de Schotten, who worked here, as Marcus said, for many years. And I will start with this because he used this slide at the beginning of one of his talks a few weeks ago for the habilitation to direct research in Paris. And basically, he started by saying, I paraphrase, every day when we start to work, when we start to do our job, we take one or two badges that qualify us as belonging to one or two schools of thought. Localizationist or associationist, in the sense that if you are more on this school, you think that what is relevant when you study the brain is where a certain function is located, what is the origin of a certain dysfunction in a certain particular psychiatric or neurological syndrome. So where is the crucial question? If you are belonging more to that school of thought, you will think that actually brain functions and dysfunctions actually emerge from the interaction between different brain regions. It was an interesting introduction, and then he went on by discussing all of his work about brain connectivity in the last years. And then at the end of the talk, was Masoud Hussain from Oxford, and he made a very, very interesting objection. He said, okay, you propose us that these are two very different ways of looking at the brain, but in order to look at connectivity, you need to define the seed of your connections. If you don't have this, you cannot have that. And that was a very, very sharp, not objection, but observation, because it's something that we do not think that much about. I mean, it's not really in opposition between the two. So, and indeed, it is a very, very hard problem, because how would you define the seed for your connections? Most likely, the first answer that comes to your mind is, okay, I will use some macroanatomical landmark. This can be a good idea in some cases. It can be a very bad idea in other cases. You know, all of you know that, for instance, the border between primary sensory and primary motor area is in the anterior bank of the central sulcus, but it doesn't work like that for other regions, and especially for higher order cognitive regions like in the Broca's region. Here you can see that actually you always know that Broca's region is in front of the presental sulcus, in the inferior frontal gyrus, but subdivision between the A44, 45, and 47 does not follow the sulcar anatomy. The ideal situation would be to have, to be able to extract differences in site architecture directly from our in vivo T1 weighted image or whatever kind of image. Unfortunately, this is not possible because actually the T1 weighted signal is very insensitive to the site architecture. Truth to be said, I mean, there have been a few attempts in the last 10 years, but they did not give really great results. Indeed, when you look at the T1 weighted image, the biggest contrast that you see is between white matter, gray matter, and CSF. You don't actually see differences in the layering in the cortex. Truth to be said, recently some people have proposed that although the T1 weighted signal is not sensitive to site architecture, it is actually quite sensitive to myeloarchitecture. So if we hypothesize that there is a strict relationship, a strong link, although not a perfect correspondence, between site architecture and myeloarchitecture, in the future, with very high resolution T1 images, we would be able to detect differences in myeloarchitecture in the single subject and possibly also in vivo, which what we are mostly interested into. However, this thing right now is still in its infancy. And that's the reason why, as you probably know, there are these maps which are being created by the group of Kartziles and Katerinamus, where basically ex vivo site architectonic mapping of different regions. And it takes an enormous amount of work. It would be in only 10 subjects. It will not be in the subject you use for your own experiment, but still, until today, is the best way that the most reliable way that we have, the most reliable method we have of the variability in the site architecture in different areas of the human brain. Now, you could also tell me, well, if you want to localize the seed to perform your connectivity analysis, you could just use a very simple, not necessarily simple, but a very used functional localization method like fMRI. You could run a certain localizer, and then you could look at the interaction between different nodes that you identified with your localizer. This is what has been done in many studies. Typically, this is a study from 1997, very beautiful one. Two different tasks. Pay attention to rotating configuration of dots, or just watch it. Pay attention to whether it changes the speed. With normal GLM analysis, you find exactly the same regions. So in this case, the simple GLM failed to find the difference between the two tasks. But then, if you look at the simple methods like structural equation modeling, you find that actually the influence of one region on another changes according to the task. So, to sum up, you want to define your regions before doing the connectivity analysis. You can rely on macroanatomical landmarks, not a very good idea unless you're looking at primary sensory motor regions. You cannot rely on site architecture because you cannot extract this from your MRI images. You may be allowed in the near future, near or distant future, to use mild architecture. For the time you cannot. You can use functional localizers, but these also have some limitation in the sense that first, you need to have the same paradigm in all the subjects in which you want to analyze the connectivity. And second, of course, well, your connectivity analysis will be limited to the paradigm for which you acquire the images. You cannot analyze connectivity between regions that do not come out from your fMRI localizer. You might also have problems of thresholding of the fMRI stat images. You may not assume that there is a direct anatomical connection between all the nodes. So it's a very great method, but you have limitations. And this is where the studies that I've been doing in the last years come in. So let's think about the problem again. Because we said, as long as we described it until now, the task of analyzing the connectivity between different regions has a particular sequence. First you define the regions, then you look at the connectivity between them. The question is whether it is possible, whether it would be possible to go in the other way around. I'm sorry, it's really warm inside here. To go the other way around. And what do I mean by going the other way around? Not by localizing our regions and then looking at their connectivity, but starting by their connectivity to find the seeds, which looks very counterintuitive, but I want to explain a little bit better what I mean by that. There is one statement in, there is a sentence which was written by a very famous neurologist, Mesulam, in the year 2000, or maybe 1990. It says, nothing explains the function of a neuron better than its connection. And of course, you can replace neuron with brain region, most likely, or even brain network. So, I think it's a very, very sharp and interesting observation because it's very simple. And when people make very simple statements, they let us think about something that we give for granted, so maybe they give us new insights. What this sentence tells me is that if, indeed, the connectivity constrains the function of a certain region and other anatomical properties of that region, then it should be possible to start by looking at the structure of the connectivity of a certain region and then recover its functional and anatomical properties. And this is the kind of thing, the kind of approach that I tried to give some contribution in the last years. So basically, you look at a relatively large region where you think that there are different functionally or anatomically segregated regions, or where there is a certain property, anatomical properties, which flows from one to the other side of that region. And then you use some particular methods to recover the location of the different regions that you hypothesize that are there, or to recover the trajectory of variation of a certain anatomical properties. I will give you an example, very, very basic example. The thalamus. Very nice structure. We know that the thalamus is composed of many different nuclei. But if we look at them, if we look at the thalamus in T1 weighted image, we see something which has the shape of an egg. We cannot really distinguish the different nuclei. We can maybe say, okay, I think that the pulvinar is around here. I think that you can maybe see where the geniculate nuclei are, but you cannot really distinguish one from the other. You cannot say where the border one is with respect to the other. Crucially, however, each one of these nuclei has different connectivity. So for instance, the medial dorsal nucleus of the thalamus, which is the one that increases more in humans with respect to other primates, is directly connected to the prefrontal cortex, and so on and so forth. The different geniculate bodies are connected to different brain regions, primary auditory and primary visual, and so on and so forth. So the point is, if I would have an information about the whole brain connectivity of each voxel in my thalamus, and if I have an hypothesis about where different regions of the thalamus actually project, I could use this information to estimate the probability that a certain voxel has more connection to one of these targets with respect to the other ones. And this is the seminal work that was done in this kind of contextual studies, which would be called generally connectivity-based classification. So basically, you have the whole thalamus. You don't put any a priori assumption about the location of the nuclei. You seed your tractography, in this case, from every voxel of the thalamus to a certain predefined amount of targets, and then you look at the probability that each voxel in the thalamus is more connected to one than to the other targets. And in this way, they recovered a map of the thalamus which actually resembles, to a certain extent, to the maps that you can get by means of histological examination. Very nice work.
I have some concerns about these.
Go ahead. Well,
Perhaps
we all
we should
have,
leave
but,
it. We should leave it at the end. If
as you
we're
prefer.
gonna return.
You can go ahead now if you want.
Well, there's obvious limitations of this approach. It was based to the probabilistic tracking they did was affected by the distance. So you could almost predict a priori that would be the map generating the thalamus, because by simply looking at the distance between those cortical nodes and this thalamic nuclei.
Yeah.
And indeed, you know, I think, you know, for example, they never separated the projection of the medial dorsal nucleus, which we know exactly goes medially, from the rest. So this is a big area, the kind of the purple one. Because that's where they had problem. So I think it's a bit misleading in that sense, and the thalamus is not the right example to use.
They chose the thalamus because of what they described as a particular penetration of white matter inside the structure. Of course, there are limitations due to the distance, limitations that actually cannot be accounted properly even now. And it was interesting that the result was relatively similar to what they were expecting. Of course, it's a work of 12 years ago, and yeah, it has limitations that, to a certain extent, they tried to account in recent years.
So look, for example, they had a lot of frontal connections coming from the metathalamus.
Yeah.
You know, that's where the medial geniculate nucleus and the LGN comes from.
So it's definitely not perfect. I mean, it's something that at that time was something quite amazing, I think. Of course, there were limitations, but I think that at that time it was something quite amazing.
Okay.
Anyway, let's go on. Let's go on, because I mean, all of these works that I'm presenting to you, of course, they are not free of limitations. The idea is to see where did it start from and where it is going. The one limitation of this work, in terms of a hypothesis, is that you didn't have a particular assumption of the seed, but you had still assumption of the target. Because you could describe the target in a very, very simple way. I mean, prefrontal cortex, temporal, and so on. I mean, they were basically overlapping different lobes of the brain. If you take a region which is more complex in terms of the differences in the projections, like the medial prefrontal cortex, so F3 and F6 in this drawing, in the macaque, which is SMA and pre-SMA, there you can do something different, because here we know that actually they have different site architecture, different function, different connectivity. but it is quite difficult to define all the different targets of these two regions. The spectrum of projections of these two regions is quite extended. So what you can do here is instead is something relatively different. Instead of looking at the proportion of samples that go to one or the other region, you can basically look at what we call the connectivity profile. That is a vector that tells you for each voxel in the brain whether there are samples coming from a certain voxel in the seed region. When you have this kind of information, you will have a big matrix that tells you for each voxel in the seed region where are the orthography samples located in the rest of the brain. And at that point, you look at the differences within the seed region. You look at, for instance, how they are similar to each other, how each connectivity profile is similar to each other connectivity profile. And at this point, you can use a correlation or distance measure to establish this. At this point, you can use a certain clustering method, for instance, to recover different regions in that particular seed region. So in this case, you can actually recover SMA and pre-SMA to a certain extent of precision. This is not the original work where it was published, but it's a second work which replicated the results. I show this because there is also a nice indication about how you go on in this particular method. Recently also Michel produced a parcellation of the whole occipital lobe, and the interesting thing here is that he used the clustering technique but without having to input manually the number of clusters. But this is a very, very different and open story. I cannot talk about that now. Now, so far I didn't show you anything about my works. Now here we go. The idea is these methods can give us some interesting result when we expect a very sharp transition of the connectivity between the clusters, or when we expect clusters to pool. What if we look at a certain region of the brain where we cannot expect clusters because all the anatomical texts tell us that there are no sharp transitions in connectivity, cytoarchitecture, or functionality? And this was the problem that I had with this region, the insula, that probably some of you already encountered in your work. One of the problems of this region is that it is involving a tremendous amount of tasks, which means probably it is not specific to any of them. And indeed, although there is a high heterogeneity of site architecture, function, and connectivity in the insula, it appears that there are no sharp transition between the different subdivisions of the insula. And this is true whether we look at the connectivity or whether we look at the site architecture. Plus, it looks like in this case, in the case of the insula, the macroanatomical landmarks are of no use at all to define different fields, the different cortical types of the insula, because they are basically perpendicular to the macroanatomical landmarks. So what do you do here? It's a big problem because you cannot use clustering techniques. If you would use a clustering techniques, I can promise you would get some clusters. The question is whether the result that you have from your technique reflects something which is actually there in your data. If you use a clustering technique in a region where you would not expect a sharp transition of the anatomical property of your interest, you would still get the clusters, but it would be a really nice and big artifact. So what we did first was to test whether our hypothesis about the absence of sharply segregated clusters was actually plausible. It's still good to test whether your ideas are legitimate. So basically what we did was to use actually a clustering technique, but we repeated the clustering not one, but 1,000 times. And the idea was that if there would have been a very clear segregation between 2 or 3 regions in the insula, we would have had a very low uncertainty in the determination of where these regions are. So basically, we would have seen that all the boxes in the insula, or almost all the boxes in the insula, would have been able to be classified into 2 or 3 different, very clearly distinct clusters. This is not what happened. We saw a very large part of the middle insula which was actually impossible to classify even in the single subject. We saw the same thing happening, interestingly, also in the medial prefrontal cortex. However, crucially, the amount of voxels in the insula that could not be reliably assigned to 2 or 3 clusters was much higher than what happened in the medial prefrontal cortex. Now, this told us that most likely we were right about thinking that the insula could not be subdivided into clusters. This still didn't tell us what kind of structure or connectivity we would have in the insula. That's why we had to use something different. The right way of doing this is to use a method that can actually tell you what kind of structural connections you have in your region of interest, that is able to detect whether there is a structural connectivity at all, whether this structural connectivity can be described as clusters, or whether it can be described as a gradual transition. And this is why, after looking for a long time, we came up with a method called Laplacian eigenmaps, which is a spectral method. I can give you the details later. The greatness of this method is that indeed it doesn't assume a specific structure of connectivity, but it can detect different structures of connectivity, and therefore it is much less prone to artifacts. In this way, we were able actually to detect that indeed in the insula there was a gradual transition of the connections, while in the medial prefrontal cortex there was a sharp segregation of the 2 clusters. And of course, when we compared the trajectory of connectivity variability that we found with the projections in the brain, we found that it was relatively consistent with what we would expect from the tracer injection studies in macaque. Now, this paper was published in 2012. Fortunately, from that time, somebody read actually the paper. Actually, a friend of mine was there in Groningen. And he actually used it for other studies, this time using not anymore tractography, but functional connectivity. And found interesting results that he presented last year and this year in the OHBM conference. So here, basically, he looked at the functional connectivity of the entire placental gyrus, and he found a dorsoventral mode of connectivity organization which goes by itself, but most likely refers to the somatotopic organization of the primary motor cortex. Interestingly, if I would have to choose something to look at the somatotopic organization, I would have chosen S1 because there it's much more clear that there is a very, very sharp somatotopic organization, but they did it with M1 and apparently it worked. M1 and also premotor cortex. What is more interesting is the second example. Here he looked at, well, it is written primary visual cortex, but actually it includes also some part of V2 and V3. And you probably know that when you look at when you're interested in the primary visual cortex, you can do this retinotopic maps, in which you can map where the location of the fovea is with respect to the periphery, and in which part of the cortex a certain part of the visual hemifield is represented. Now, the interesting thing was here that he used these Laplacian eigenmap methods on the connectivity profiles, functional connectivity profiles of the occipital cortex, and the first mode, the first trajectory of variability of the connectivity described a map which was very similar to the one that you get when you look at the eccentricity. And the second mode of connectivity organization actually looked like the maps that you get when you look at the polar angle. So the interesting thing here was that this method was able to recover 2 different structures of connectivity overlaid onto each other. And I think that this is extremely...
Leonardo, could you explain the first mode and second mode? What's the difference? I didn't get that.
Okay, I will make a very simple, hopefully very simple example. How many of you have used the principal component analysis here? PCA.
Pass.
Becomes difficult.
Simpler than that. Simpler than that.
It becomes difficult. It cannot be simpler than that. I mean, how many people among you used ICA on resting state data?
Pass.
Okay, let's put it in this simple way. When you... Okay, let's put it in this way. No. How many of you used multiple regression? Come on, no. Multiple regression. Multiple regression, guys. GLM, fMRI stuff.
We use it, but we're not aware of
Good.
it.
Okay. My God. Okay. You have your imagine your model in your GLM. What is the most important requirement of the predictors? You have two predictors in your model. Let's say that you make an fMRI experiment where you ask the subject to either do some finger tapping or some movement with the foot. What is the most important requirement, not just for fMRI, but for any general linear model where you have different predictors? When you have predictors which are not just one, but 2, 3, 4, as many as you want. What is the most crucial thing that has to happen? How do these predictors have to look like?
Should be independent and correlated.
Yes, exactly. And so independent is one thing. As far as I know, for GLM, for general linear model, you just need that to be orthogonal. At least this is how I conceive it. So orthogonal, it means that the variability which is explained by one predictor cannot be confused with the variability. It is difficult. It means that there are two sources of variability which are independent from each other, and that's why you design your predictors in this independent way, because you make the work more simple for your GLM, not to confuse between the two predictors. So this is also how PCA works, in the sense that you extract different components telling you about the trajectories of variability in your data set, but they have to be independent. Otherwise, you will have some big problems. This is what happens here. You have a total amount of variability, which is your big matrix of functional connectivity, which is decomposed into two sources of variability. One which refers to the eccentricity organization, so the location of the functional connectivity with respect, the map of the functional connectivity with respect to the location of fovea and periphery, and another source of variability which is mixed with the first one, but it is unmixed by the method that you used, in this case PCA, Laplacian eigenmaps, ICA, whatever you want. In this way you can decompose these two sources of variability, and in this case these two sources of variability correspond to two different functional properties of the visual cortex. I hope that the comparison with the multiple yeah.
Do you need to specify something to get it or actually too strong?
That's of course I expected this question, and it's a very good question. Like in almost every dimensionality reduction technique, you have always the problem of whether you have to specify or not how many components you want. And in the case of Laplacian eigenmaps, you do not have to specify it in the sense that, in principle, this is a pretty technical thing, but to answer to you, Flavio, you have as many components as the dimensionality of your connectivity matrix, basically, like in PCA. And then you can look at how much variability you explain in the first third, second third, let's say first 10 components. Of course, you have to choose where to stop. But in principle, you don't. It is not like in clustering. In clustering, you have to say, I want 2, 3, 4, 5 clusters. No, here you don't ask how many components you want, how many modes you want to look at. That's something that you do not explain in the poster in which it was published. But that's a very important question, actually. I mean, they report 2 modes. How much of the variability, of the total variability they explain, that is not. It's not, but it's a very good question. Actually, I should ask it to Kuhn.
And Leonardo, these are tractography based.
No, these are based on resting state functional connectivity.
Resting state.
Yeah. Yeah.
Okay.
Yeah. I'm sorry that I didn't
Okay,
specify.
because I thought it was you were coming from Insula, which was a structural. Which
I'm
was
sorry.
Tractography. So I assumed that this was tractography.
I'm sorry, sometimes
No, it's
I
resting
forget to
state.
specify. Yeah,
Okay,
yeah, this
okay.
is an absolute resting state. I'm sorry, sometimes I forget to specify because in general, these methods can be applied to both structural, to both anatomical and functional connectivity. In the past, they were more used for tractography. Nowadays, people are starting to use them more for functional connectivity or do a mix of them. But in any case, this is resting state functional connectivity. The work on the insula was with tractography. There have been also works where they tried to parcellate the insula based on functional connectivity, but they were using clusters. Anyway, they found some results, but whatever. I think that we demonstrated that there are no clusters in the Okay, last example of this kind using notation again maps. Well, it's an example related to the basal ganglia, actually to the striatum, not the entire basal ganglia. With the same method, what they did was basically to find different modes in basal ganglia, and also in this case it's resting state functional connectivity. You know that the striatum has different functional organization, in the sense the dorsolateral striatum is more related to motor functions, basic motor functions, inhibition or disinhibition of premotor and primary cortical areas, primary motor areas. Central striatum has more to do with central executive function and projects direct to the prefrontal cortex, and ventral striatum is more related to emotional and motivational tasks. It has a strong link with the limbic system, and for instance, you know that nucleus accumbens, which is indeed in the ventral and medial striatum, has a strong link with addiction. So they applied the same method, the Page-Hagen maps, to the connectivity matrix of the entire striatum, and again they found two modes. One which describes the main subdivision of the striatum, so basically putamen, caudate, and ventromedial striatum, sharing core, and the second one which was actually consistent with the predictions in terms of connectivity. So this is just another example about the fact that also in this case there were two overlapping trajectories of connectivity variability, each one of them having a different anatomical or functional meaning or interpretation. But this work that I will not discuss because it's actually already quite late. So, summary of this part. I'm anxious to go to the next part, and probably also you. Summary of this part. We can actually study the structure of connectivity of an entire region and try to recover some properties of the subregions composing that region, or about a particular property flowing in the entire region and changing gradually from one to the other side of that region. So we can look at different structures of brain connectivity, both clusters and gradual transitions, and probably other ones. We can see that we can recover trajectories of connectivity variability which are related to the functional specialization of different regions. Sometimes we can recover overlapping modes of connectivity variation, and then the last thing, as I specified now, usually you can use the same method for functional connectivity and for anatomical connectivity. Before going to the next slides, somebody has something to ask me about this part?
Has anyone looked at the multi-layered structures with the actual anatomy, or with the
With the actual anatomy, what kind of data?
or the underlying connections?
Well,
It
what
would
kind
have to be tractography.
No, it doesn't need to be tractography. I mean, it needs to be whatever kind of data you can recover for a voxel, there it is multivariate. I mean, if you look at the T1 weighted images, each voxel has a value, and that's it. Of Of course, you can still do connectivity analysis in that case because there is this way of analyzing which I don't remember exactly the denomination right now, but basically you can treat a bunch of subjects and for instance, gray matter value in each voxel as a multivariate measure of the possible variation that the possible variability that you can have in T-automated measurement in that particular voxel. I don't remember how is that called, but in principle you need to have a measurement for each voxel which is divided in terms of many different numbers, say. So it's connectivity, or I don't know, it could be also different properties extracted from the diffusion weighted signal, for instance, or functional connectivity. And the structural, I don't know, but we can talk about that. That would be extremely interesting, I think. Well, there is an example of that actually in the cytoarchitectonic maps of Ziles, because there they have such a high resolution that basically the way that they find the borders is by defining each. I don't want to say column because it's not exactly this, but you have the cortical ribbon here. You define it in many different rectangles, and for each rectangle you can quantify different. The value of your staining at different locations in the rectangle. And then you can calculate different statistical moments, and at that point you have a vector of values for each little part of the cortex. And then you can look at distance between two adjacent rectangles or sets of rectangles and find these boundaries. This is how actually they create the cytoarchitectonic maps. So in that case, you can do it. If you would have very high resolution T1 weighted images, probably you could do something like that. But otherwise, I don't know whether it would be possible. Let's go to something completely different. Now, we already mentioned anatomical connectivity. Now let's go totally into functional connectivity and specifically to resting state functional connectivity. In general, do you know what is resting state functional connectivity?
We had a talk, I think last week, from Caramilla, so...
Ah, okay, well, then I can skip some stuff. I just want to tell you that it's a very interesting effect that was discovered in 1995 by Bart Wieswald with a very, very simple experiment. It was basically asking to a person to go into the scanner, stay there for a few minutes while he was acquiring fMRI signal, without any particular task. Afterwards, he asked to stay in the scanner for another 5 minutes and he asked to do a finger tapping task. He looked at which were the regions in which the finger tapping task was correlating, the activity was correlating in the brain, and he found a map like this. And then he took the same seed regions and he used them as predictors of the resting state activity in the rest of the brain, in the resting state data, and he found basically the same activations. This was a very, very interesting, well, very surprising result actually, because if you expect that in the resting state signal there is only noise, you shouldn't be able to find a very well localized activity which is consistent with the activity that you would have if you ask the person to do finger tapping or whatever other task is usually associated with the activity in that region. This happened in 1995. For a lot of time, people didn't give a damn about that. But after a while, people started to see that actually this kind of resting state networks were present not only in the motor system, but also in many other parts of the brain. And this was very interesting, but mostly something like an old curiosity shop thing, until somebody showed that actually if you look at the spatial distribution of these resting state networks, you can see that it's very similar to the distribution of the networks that you would have by using task-evoked activity. This is a work of 2009 where a particular method called ICA was used. And the ICA was fed with either the resting state data or the data from a database of 1700 studies where the activations were basically the locations in the brain where specific tasks were producing a specific activation. The result was quite striking because the maps were looking extremely similar to each other. Now, this is usually what people remember about this paper, the fact that maps are similar. The important thing, another important thing, is how these maps are actually extracted by means of ICA. Okay, ICA actually gives us two information. One is the spatial map. The other one is the thing that I was most interested in. How do you think that these spatial maps are actually produced? I mean, why does this method yield to these spatial maps? I mean, what is the defining feature which makes so that all these boxes are grouped together in one component, and not in this component, all these boxes are grouped together in this default mode network, and so on and so forth? Why is it so? Why there is this subdivision?
It's a temporal correlation.
Exactly. And this is something that usually people forget. ICA gives you two information. One is about the spatial distribution, which is the feature for which spatial independency is optimized. The other one is the temporal information. It means that basically, even in resting state signal, voxels that have similar temporal, similar time course in time are grouped in the same spatial component. So for each one, for each spatial map, you have characteristic time course, which is describe the mean resting state activity of that particular component. This is interesting because it was already known from about 2005 that different networks can have different degrees of correlation with each other. In the sense that, for instance, here you have default mode network and so-called central executive network or task active network. You know that the central executive network is usually active when you're doing something very stressful, when you're doing mental arithmetics, when you're doing n-back tasks. Default mode network is exactly the opposite. It is the network which is more active when you're resting, when you're relaxing, thinking about your next vacations or your previous vacations, even getting bored. So basically, they have, behaviorally speaking, an anti-correlation with each other. It is interesting that this anti-correlation, this negative correlation, sorry, not anti-correlation, this negative correlation is also found in the resting state signal. So in resting state signal, when the one goes up, the other one goes down, and so on and so forth. The question is why there is this temporal structure. I mean, why there should be any differences in the resting state signal of different networks. Why it should be so? Somebody said the resting state signal is simply the residual activity, which is the residual noise which is derived from the fact that these regions are anatomically connected. If it would be so, why there should be any difference in the temporal, in the time course of activity of these networks at rest? As many interesting questions, actually we don't know why there is this difference in the temporal structure of different resting state networks. It is possible to hypothesize, well, in the case of the synchrony, it is actually easy. Regions which are distant in the brain, but whose bold effect goes up and down together, are most likely, are very likely to be active at the same time for the execution of a certain task that requires the activity of both of them, that all the regions of the network. For the anti-correlation, it's actually more difficult. We can hypothesize, but this is just an hypothesis, that this contributes to the functional segregation between different networks, in the sense that we can hypothesize that if different networks have different time courses, they can actually activate each other without interfering with each other. So in a certain way, the difference in the time course between resting state networks could relate to the possibility of having a flexible and smooth interaction between them without at the same time having interferences. This is the hypothesis that I use to look at something that many of you probably encountered in your study, which is autism. So keep in mind what I told you, the fact that there is a certain temporal structure in the time course of the resting state networks. I use this to look at autism because if we look at the symptoms of autism, we can see that they are very heterogeneous. There are problems in linguistic and social interactions, stereotype and repetitive behavior, and then some problems with the sensory processing, atypical sensory processing. One thing that many people propose that this heterogeneous list of symptoms have in common is the fact that they do not refer to basic brain functionalities, but brain functions that actually require the integration of different regions, of the activity of different regions, or the integration of the activity of different networks. If you think about social interaction, it's probably the single human activity which requires the widest interaction between different functional networks. It requires theory of mind, mirror neuron, language understanding, language production, autobiographical memories, saliency detection, and so on and so forth. So you need to keep in balance the activity of all these regions and all these networks. Indeed, it has been proposed that if autism is considered as a problem of functional integration, a possible substrate, anatomical substrate for this could be the presence of an excess of local connections coupled with a defect in long-range connections. Now, it is true that indeed in the last something like 15 years, many work that look at problems of functional integration in autism found actually quite interesting evidence to support this hypothesis, to support the hypothesis that autism can be studied as a problem of functional integration. I will not go through all of these evidences, but because there are many, but the idea here is that you can actually find problems of functional integration at many different levels of anatomical organization. Now, what we did was to focus on two levels which were still not studied, and I will talk to you especially about this second level, about interaction between networks. The hypothesis here was that indeed, if the temporal structure of the brain networks, of the resting state networks, is something that actually contributes to a smooth functional integration of different brain networks alteration of this temporal structure could be present in autism. And here we'll talk about high functioning autism. So what we did was basically to extract our resting state functional network by means of ICA, actually in this case by means of meta-ICA, which gave us more clean networks, and then look at the temporal structure of these resting state networks. What we did was to look at that in the single subject. So in the single subject, we look at the correlation between the activity of each network and each other. And then we look at how the average activity in one group would be different from the average activity, the average correlation, in the other group. I think I will go direct to the results because I see it's actually quite late. So, what we saw was quite interesting. Many studies about functional connectivity in autism have shown a decrease of functional connectivity, for instance, between posterior and anterior regions involved in language production. What we saw was quite different. We were expecting that, but we saw something much more specific and very different. We saw actually that in the sample that we used, and it was a large sample, it was something like 359 participants, we saw a consistent increase of the resting state functional connectivity between subcortical regions and a set of other regions. keep in mind that we looked at the interaction between 19 different networks, and these are the only solutions for which we found a significant difference between the two groups. So it was very, very specific. So there was a hyperconnectivity, if we can call it like that, increased interaction, altered temporal structure of the interaction between subcortical nuclei, basal ganglia and thalamus, and some other regions, most of them including primary sensory cortices. So here there is primary auditory, ventral somatosensory and motor cortex, dorsal somatosensory motor cortex, and V1. This was a very intriguing result because, as you already know, in autism, the academic literature focuses mostly on the social problems, while it is already 20 years the clinical literature stresses a lot the presence of sensory symptoms in autism. And actually only recently, only last year, or 2 years ago probably, the DSM-5 finally included the category of hyperreactivity and hyporeactivity as defining criteria for the diagnosis of autism. In our case, we basically said, okay, this must have something to do with sensory symptoms. We also saw a correlation with autistic traits measured by means of SRS, and we thought, okay, basically this would be interpreted in terms of an excessive predominance of basic information processing in the brain of autistic people, which on the other side would bias processes devoted to detecting saliency towards sensory elements in the environment. I am almost done. I showed you something which relates to tractography, something which relates to functional connectivity. I tried to show you some ways in which you can actually study the structure of anatomical and functional connections. I want to show you just 3 more slides, and I will go very fast, that show what could be done in the future with this kind of methods to extend basically the range of possibilities of these methods. Because so far, especially for connectivity-based parcellation, we've seen that, okay, now we can actually segregate different regions with different potentially cytotectonic properties. Okay, but once we have that, can we do something more with respect to that? So the first study that I want to show you very quickly is this one. It's from 2009, so it's quite old, and it's quite interesting that nobody used this method afterwards. But basically, here the authors first made a classification of the entire basal ganglion by means of cartography, so connectivity-based classification. Of the striatum again. And then they looked at the relationship of the connectivity strength of specific regions with some personality traits. And specifically, they looked at relationships between knowledge-seeking and reward dependence with connectivity with the amygdala and the hippocampus and other regions in the cortex especially. For reasons that I will not stay here to tell you, they found that the strength of the connection between the ventromedial striatum and the amygdala and the hippocampus was particularly strong, and the strength of this connection was particularly correlated with novelty seeking, while the strength of the connection of the dorsolateral striatum with other cortical regions was particularly stronger for reward tendency. Irrespectively of this particular study, the interesting thing here is that they were able to define very clearly some regions from which they could see the tractography and look then at the relationship between personality traits and connectivity strength. The concept of connectivity strength is of course quite questionable, but it is just an example of how the results of tractography could be used. This is another example from Michel. It's a work that is submitted recently. And basically here there is a parcellation of the frontal cortex, of the entire frontal cortex, which is already per se quite interesting. Nobody did it so far. And there is a relationship between the parsers that you find here a certain relationship, although not a perfect correspondence between, well, perfect correspondence nobody can know, but some of these parsers resemble to distinct cytotectonic regions, some others actually do not. The interesting thing here is, can we get some other information besides saying, okay, these parsers belong to different regions with different cytoarchitecture? He tried to look at the relationship between these subdivisions and other properties of the brain, and he saw, for instance, that these regions have an increasing cortical thickness, and that they have an increasing entropy when we look at the resting state signal inside each of these clusters. So, in this case, he basically showed that you can use the result of the parcellation to look at other properties in the brain, functional or anatomical properties, and try to establish some link with behavior problems. Very last slide. Beautiful study from 2012, which goes back to the very beginning. So we started with the idea, okay, is it possible to start with the connectivity and retrieve some functional properties of the regions that are connected to each other? Well, this study exemplifies in the most beautiful way that it is actually possible. Because what they did here was to make a very simple fMRI task-based experiment in which they showed some faces contrasted with some natural images, with some landscapes, to a set of participants, and they recorded the fMRI activity, which is here. And then they looked at the anatomical connectivity of the same voxels, and they tried to model the result of the fMRI experiment by means of the structural connectivity. At this point, which is exactly what you do in your GLM, you have your Y, your expected, your acquired data, you have your predictor, and you have a b-value, either 1 or a vector of b-values, that model how your model can actually use to predict your data. After having gotten these weights, so basically how do we have to weigh the anatomical connectivity to get to the fMRI data, they use it on a different group of subjects. And in this way, they were able actually to predict the fMRI activity just by looking at the anatomical connectivity. And this is the result. So this is the actual results of the fMRI experiment, where they were shown faces and natural scenes. And this is the prediction of the fMRI result made entirely on the basis of connectivity. So, conclusion, yes, most likely the information that we have when we look at connectivity, even just structural connectivity, is a quite interesting information that can actually predict the functional properties of different brain regions. I think I will stop here and listen to your questions if you have any.
Right, okay, so time for questions. If people have to leave, please leave now, because I guess the question will go on for about 5-10 minutes. So if you want to leave